This protocol describes how best to functionalize the AFM cantilevers using single T cells and solid particles. These tips can then be used to probe single-paired dendritic cell T cell interactions and to monitor cellular responses of phagocytes, respectively.
A main advantage of this technique is that it uses a biocompatible glue for single T-cell functionalization of cantilevers. This glue is inert to eukaryotic cells, thus keeping T-cells in their baseline activation state.
These methods provide insight into immune cell activation and complex intercellular events, such as immune synapse formation. The methods can also be extended to other systems involving cell-cell or cell surface interactions.
For someone that is new to this technique, it is important to make sure that the T cells are in good condition prior to use, and that they are pre-treated with IL-2 overnight. Additionally, when using the biocompatible glue, move quickly to protect from unnecessary oxidation.
To begin this procedure, prepare the single T cells for microscopy by following along in the text protocol. Then, prepare glass cover slips seeded with DC 2.4 cells, and incubate the cells overnight in a humidified chamber at 37 degrees Celsius and 5% CO2. Clean soft, tipless cantilevers with a low spring constant using piranha treatment, plasma cleaning, or UV ozone cleaning. Then, mount the cleaned cantilever to the AFM scanning head.